Welcome back to Mitochondrial Medicine 2021. We continue now with a clinical trial session featuring Gensight Biologics, Minovia Therapeutics, PTC Therapeutics, and the University of Florida.
Hello, everyone, and welcome to the clinical trials update portion of our programming at Mitochondrial Medicine 2021. In this next session, we will begin to hear clinical trial updates from representatives of both academia and industry through a series of panel discussions. I think you'll find them very interesting. And in this first panel, I'm joined by representatives from Gensight Biologics, Minovia Therapeutics, PTC Therapeutics, and Taiel Pharmaceuticals. Our first panelist is Dr. Magali Taiel, Chief Medical Officer at Gensight Biologics, and she will provide an update on the company's ongoing gene therapy development for LHON. Dr. Taiel.
I would like to warmly thank the organizers for the invitation to present a few insights of our work. Firstly, one word on Gensight Biologics. Gensight Biologics is a biotech company developing gene therapy in inherited retinal diseases leading to blindness. We have two main projects using two technology platforms. Our lead program is the development of a gene therapy in LHON. We have another program in retinitis pigmentosa, and we are at early stage clinical development with a first-in-human study which is ongoing. Gensight was founded in 2012, and the headquarters is based in France. I would like to focus this presentation on Lumivoq gene therapy development in LHON. Let's move on to the disease. LHON is a maternally inherited mitochondrial disease. It is a rare disease with an incidence of 1,500 new patients per year in Europe and in the US. Before onset of the disease, the carriers of these mitochondrial mutations have a very good vision, 20 out of 20. One day, subjects have a dramatic drop of vision, firstly in one eye, and a few weeks later in the second eye. You see this drop of vision on the left graph. Rapidly during the acute phase of the disease, these subjects reach their lowest visual acuity, which is called the nadir. This is typical of this disease, a rapid and sequential drop of vision. Within 1 year, the patients become blind. The onset of vision loss is on average between 15 and 35 years old. It mainly affects males. Different mutations on the mitochondrial DNA can lead to LHON. The 11778 mutation on the ND4 mitochondrial gene is the most frequent mutation, representing around 75% of LHON in Europe and in the US. It is also the mutation causing the most severe clinical form of LHON, and Lumivoq is specifically targeting this mutation. Importantly, LHON is a neuroophthalmic disease, and such as glaucoma and many neurologic diseases, anatomic modifications start before the occurrence of symptoms. In LHON, the retinal ganglion cells and their axons are affected, and on the graph on your right, you see that the retina observed at OCT presents anatomic changes starting before the occurrence of vision loss. There is firstly an increase of thickness of the retinal nerve fiber layer related to edema, and then a dramatic thinning of this layer over time. This is a representation of what LHON subjects face during their daily life. Clinically, this manifests with central vision loss, which is the hallmark of LHON. They get a central scotoma, which rapidly enlarges and becomes very dense, with a dramatic impact on their quality of life. We developed the technology of gene therapy to overcome genetic mutations in the mitochondrial DNA. The gene therapy Lumivoq specifically targets the 11778 mutation on the ND4 mitochondrial gene. Lumivoq is a recombinant adenovirus type 2 containing the wild type, the healthy mitochondrial ND4 DNA. Lumivoq is injected via intravitreal injection, as it is the best way of administration to reach out the targeted cells, the retinal ganglion cells. Once injected in the eye, the ND4 DNA is transcribed into mRNA in the nucleus. Then the two mitochondrial targeting sequences actively shuttle the ND4 mRNA to the mitochondrial surface, where a functional ND4 protein is synthesized and translocated inside the mitochondria to restore energy production. What are the key milestones for Lumivoq? We filed for EMA registration in September 2020 based on the results of 2 pivotal studies. We will then be moving to BLA for US registration in Q2 2022 based on the results of a third pivotal study designed with the FDA.
Thank you, Dr. Taiel, for that update. Our next speaker is Natalie Yivgi Ohana, CEO of Minovia Therapeutics. Natalie, please give us an update on your clinical trial activities.
Thank you, Phil. So in Minovia, we are developing mitochondrial augmentation therapy, which is a cell therapy to tackle mitochondrial diseases. And our hypothesis is actually that a multisystemic and complex disease such as mitochondrial diseases requires a systemic and complex modality such as a cell therapy that we are in Minovia are developing. Hematopoietic stem cells, HSPCs, are an appropriate target from our side. They are long-lived and they are the precursors of all blood cells, including all immune cells, and they can circulate and home to all tissues or organs. So we are counting on the ability of mitochondria to re-enter cells. So we are actually enriching cells with mitochondria and on the ability of cells to transfer mitochondria between cells in the hematopoietic compartment. Mitochondrial augmentation therapy is composed of 4 steps. One is the production of healthy and functional mitochondria. And then we are collecting hematopoietic stem cells from the blood of the patients, enriching them with the healthy and functional mitochondria. And then we are infusing the cell back to the patient. We are currently in a phase 1/2 trial for Pearson syndrome, an ultra-rare pediatric mitochondrial disease caused by deletion in the mitochondrial DNA. And we are currently finalizing the evaluation of the results of these five patients that were dosed under this phase 1/2 trial. And we are aligning with the FDA towards the next trial, which hopefully will be a pivotal study. We also had initial interactions with the FDA on our future indication, which is tRNA mutations, including MILAS and MERF. And this is currently in preclinical development. And by mid next year, we hope to enter into clinical trials in this indication.
Thank you, Natalie, and really exciting possibilities for our patient population based on mitochondrial augmentation. We look forward to continued updates in the future. All right, our next speaker is Dr. Matthew Klein, Chief Development Officer at PTC Therapeutics. And Matt will provide us with an update on vatiquinone, perhaps still better known in our disease community as EPI 743 and now PTC 743. Matt, turn it over to you.
Great. Thank you very much, Phil. And thank you for this opportunity to share an update on our clinical trial of PTC 743 in mitochondrial disease. First, just a brief introduction to PTC Therapeutics. PTC Therapeutics is a biotechnology company. It focused on the development of drugs for rare diseases. The company was founded in 1998 with that specific focus of developing innovative therapies for rare diseases, and we remain committed to that purpose today. We have programs at varying stages of development. We have a robust research organization, development organization, as well as a commercial organization. This slide summarizes our different scientific platforms and research areas, including work in Duchenne muscular dystrophy, spinal muscular atrophy, PKU, as well as oncology indications and an ongoing trial for COVID-19. But our focus today is on our BioE platform, which is the platform that was acquired from BioElectron in 2019. And of course, the key lead compound in that platform is what, as Phil referred to, vatiquinone, previously known as EPI 743. We have 2 potential registrational trials ongoing right now with vatiquinone, one in mitochondrial epilepsy, which is the symptom of seizures in patients with mitochondrial disease, and the other in Friedreich ataxia. In addition, we have a second compound, PTC857, which is currently in phase 1, as well as a number of other compounds at varying stages of research. And we were very excited about the acquisition of the BioE platform by PTC because it really brings added muscle to our efforts to get vatiquinone approved for patients with mitochondrial disease. As I mentioned, we have an ongoing phase 2/3 trial in mitochondrial epilepsy. And again, this refers to the symptom of seizures in children with mitochondrial disease. This trial is based on a strong scientific rationale and a large volume of preclinical and clinical data. The target and mechanism of vatiquinone, that is its actions at the enzyme 15-lipoxygenase, has been associated with seizure disorders and mitochondrial dysfunction. And of course, over the 10 years of development of vatiquinone, we have collected a large amount of data, first and importantly demonstrating that vatiquinone has been safe and well tolerated in children and adults, and also relevant to this trial, that it's had an important impact on seizures in patients with mitochondrial disease, including reduction in the number of seizures, disruption of status epilepticus that was refractory to other medications, reduction in disease-related hospital days, and in some cases, in specific disease subtypes, we've been able to demonstrate a protective effect against mortality risk. The ongoing trial is a placebo-controlled trial, and we're enrolling 60 children at sites worldwide. The key inclusion criteria include age less than 18 years, having a genetically confirmed mitochondrial disease, that could be either a nuclear mutation or a mitochondrial gene mutation, and having motor seizures despite medical therapy. So those are really the key inclusion criteria. The trial is 6 months long for a placebo-controlled phase. So patients will get first observed for a month to ensure that they have a certain number of observed seizures, and then randomized to receive either vatiquinone or placebo for 6 months. Now, importantly, at the end of that 6 months, all trial participants will receive vatiquinone for at least another year. So if you enroll in the trial, even if you start on the placebo, you will get to receive vatiquinone therapy after that 6-month placebo phase. The primary outcome measure for this trial is reduction in motor seizures, and we'll also be collecting a number of other data points related to seizures, hospital visits, and quality of life. I do want to add that given the fact that we know that there's a lot of concerns about the COVID environment, even though it's improving, we have designed this trial to minimize the number of study site visits. So in other words, we tried to make it as convenient as possible for people to participate in this trial. And that's particularly helped by the fact that the primary endpoint of seizure occurrence is documented using an electronic device that we provide all participants in the trial. This study is underway at several sites in the United States and in Europe, and we're going to be adding new sites over the coming weeks. And we're really excited about having a large number of centers participate in this trial, again, to make it as easy as possible for those interested to participate in the trial. For more information about this study, we have 2 options. One is to contact medinfo@ptcbio.com. There's also a website, www.themightstudy.com, that has some additional information on the trial, active study sites. And also I want to add that we're, of course, working closely with the team at UMDF to make sure that people are aware of the status of the trial, sites that are open, and providing any information we can for anyone who may be interested in the trial. Thank you.
Thanks, Matt, for that update. And it's really great to see this drug candidate progressing through a clinical trial once again. We look forward to future updates. Our final speaker for this session is Dr. Peter Stacpoole. He is professor of medicine at the University of Florida and principal investigator on the Pyruvate Dehydrogenase complex deficiency trial for Dichloroacetate. Peter, I'll turn it over to you for an update.
Thanks, Phil. I'm Peter Stacpoole, and I am the principal investigator of a phase 3 multi-center clinical trial of the investigational drug Dichloroacetate, or DCA, in young children with pyruvate dehydrogenase complex deficiency. This is a nuclear encoded mutation associated with congenital lactic acidosis and neurological and neuromuscular degeneration. This is the first phase 3 trial of any therapy for this disease, and it represents a partnership among the 2 federal funding agencies, the Food and Drug Administration's Orphan Product Development Division, the National Institute of Health, and our 2 industry partners, Metazone Biotech and Taiel Therapeutics. It also represents, as you see from this map, the participation of multiple clinical sites and investigators throughout the country. What I show here are the locations of these clinical sites, as well as what we currently have as the number of individuals who have been randomized and are undergoing treatment with DCA or placebo in a double-blind, placebo-controlled crossover design. We have recently begun to activate a new clinical site at the University of Baylor University in Houston, and we anticipate quite a few patients that have already been identified at Baylor to be enrolled in this trial. The study investigates the effects of DCA in children who are entered into the trial between 6 months and 18 years of age. They must have molecular genetic proof of PDC deficiency. And what we are anticipating is that DCA, by stimulating the residual enzyme activity of this enzyme, both in the central nervous system and in other tissues, will maximize PDC activity in these children and improve their outcome. And the outcome is going to be monitored by a novel observer-reported outcome survey that was developed in conjunction with the Food and Drug Administration. And it allows parents to really be the primary investigators in terms of establishing the efficacy of DCA versus placebo, where they complete this brief survey at the end of every day during the double-blind phase. They upload this information, and the information really assesses at-home functionality of the patients. So this really involves a direct and very intimate cooperation and partnership with the families of these children with PDC deficiency. At the present time, we have 11 patients that have been randomized. All of them have the most common mutation in PDC deficiency, which involves the E1 alpha subunit. We have families of 12 other individuals who are in the process of being consented and randomized. And we anticipate that by the end of this calendar year, we will probably have approximately 20 to 25 patients randomized. Our goal is to recruit the necessary 30 subjects into this trial by or before 12/31/21. The double-blind phase lasts 9 months. Half that time, the patients will receive DCA, half the placebo. And so we're hopeful that we will complete recruiting and treatment of the double-blind phase by the end of 2022 or the beginning of 2023. Patients who finish the double-blind phase will be offered open-label DCA, and we'll hope to determine the efficacy outcome measure of DCA within the early part of 2023. Thank you.
Thank you, Dr. Stacpoole, for that update, and thank you for your perseverance in advancing this trial. I know it's been a long journey, and that it's really exciting to see it reach this point now with the trial open. So good luck. So at this point, we can pivot to a brief Q&A period with our panelists. And I'd actually like to start with Dr. Stacpoole, Dr. Klein. Both of you are experienced academicians, clinicians, drug developers. As I've mentioned, Dr. Stacpoole, I know you've been working on this DCA trial for many years. Maybe you could just speak briefly to the challenges of addressing a pediatric population inside of mitochondrial disease, because I know that has some unique challenges associated with it. Dr. Stacpoole, why don't we start with you?
Sure. Well, I think one of the challenges is that we can't rely on the patient themselves providing information regarding the tolerability and efficacy of any type of treatment. We have to rely on indirect assessment. Now, there are multiple ways of doing that traditionally. Sometimes those assessments are made by the clinicians who see the patients periodically. But really, the way our trial is designed is not to emphasize repeated clinic visits. What it's designed to do is to provide insight into those individuals who we think know the patients best, and those are the parents. And so in working with the FDA, we created this observer reported outcome measure as a novel efficacy endpoint metric for mitochondrial diseases. And it covers various domains involving neurological, gastrointestinal, motor function, etc., that really, I think, helps to recruit families, because they are empowered to really make a big difference in terms of the compliance and the follow-through of their children. One of the other challenges I think I should briefly mention is also that sometimes recruitment of pediatric patients is difficult because families are often reluctant to have their child experimented on, to put it bluntly. And particularly children who may, in their family's opinion, be relatively stable in terms of their health. They may have difficulty in convincing themselves that an investigational therapy of their child's disease might be a way to go. And so I think that's an important aspect that we engage in not infrequently in our discussions with family members, in terms of helping to help them understand the rationale for the study and the potential benefits therein.
Yeah, really good point. And as you well know, through our educational component of our mission, we tried to raise awareness around this topic within the patient community. This is a very understandable position. Really, the only way we're going to get to therapeutics for mitochondrial disease is to have our patient population willing to participate in clinical trials, but it's a very personal decision. And almost every program where we talk about this, we end with it's a conversation between that patient family, caregiver, with their healthcare professional about what's best for them. But you raise a very important point that one of the challenges we face is encouraging our caregiver portion of our community to actively be involved in clinical research and having their child a part of that clinical research as well. You brought up another a really important point here with the observer reported outcome. So I think this was a real contribution from your side. We're already thinking about how we can use that more broadly, right, across Lee syndrome and natural history studies, just to be able to build out that patient voice, right, through the caregiver. I think that's one that we'll see more or increased utility, right, in the future.
And Phil, one of the important points in developing that was we first canvassed over 24 families throughout the United States. And we asked them a simple question. And we said, what do you see when your child gets sick?
Right.
And we took their interpretations and descriptions of the manifestations of disease in their children. And then we created various types of clinical domains, neurological, et cetera, et cetera. And that's really what formed the basis of the OBS-RO that, as you know, the UMDF was instrumental in helping us validate that in an early study.
Right. Yeah, I'm really glad we were able to financially support that in that early stage. As I said, I think the real payoff is going to come now through this trial and future studies such as natural history and other clinical trials. So yeah, thank you, Peter. Matt, I'm going to turn it to you. We've just talked about observer-reported outcome, maybe reducing the burden on patients, patient families having to go to the clinic to participate in clinical trials. This is becoming more of a general trend, right? If there's some silver lining that's come out of COVID and this pandemic period, it's forcing all the stakeholders to rethink what does a clinical trial look like? Can it be more decentralized? Maybe you could just speak to that. I know you commented on it with your COVID studies.
Yeah, absolutely, Phil. I think, Dr. Stacpoole raised a number of important points and challenges for trial participation. Obviously, for kids who have, who are in wheelchairs or have portable ventilators and other equipment, it sometimes literally takes a village to get to a study site. It may require one parent, two parents. We've had two parents and a grandparent, a family friend. That's an enormous commitment and an enormous, resource sink, really, to just to get to a study site, just to participate in. And that obviously is a barrier to participation. And even when folks do participate, it just makes the clinical trial experience really, really hard and unpleasant. I think we've for years talked about how to address that, and the fact that we can do things remotely, avail ourselves of telemedicine, avail ourselves of home-based outcome assessment, visiting nurses, all those things. And in fact, at our meeting at FDA in September of 2019, as you recall, this came up in the Q&A. How do we do this? And I think everyone wanted to do it. And I think one silver lining of the pandemic was that it actually forced us to do it, both particularly in terms of clinical care. Patients quite simply could not get to clinics and hospitals for evaluation, and we were required to do remote visits. And the insurance companies joined in as well, supporting cross-border telemedicine, reimbursement for telemedicine, prescription renewals, all those things that seemed like really high hurdles got cleared because we had to. And the same with clinical trial implementation and as you said, decentralized trials. So I'm hoping we can harness that momentum we've been able to create during COVID. I know we're working hard on it, and as you said, it's going to be a multi-stakeholder endeavor. Companies, patients, physicians, FDA, EMA, we're all going to have to do that. But I think we need to capitalize on the opportunity presented by COVID to continue these efforts, because it's critically important. If we want to do clinical trials to truly understand how our therapies help patients affected by mitochondrial disease, understand the safety, understand the efficacy, we need broad participation. And really the way to get broad participation is to move towards these decentralized structures.
Yeah, good. Couldn't agree more. And it'll be really interesting to watch this going forward, to see how some of these mandates are adopted and implemented over time. Thank you for the comments. I'm actually going to turn now to our first two speakers on the panel today, who talked about two very different approaches to what I'll broadly describe as personalized medicines. We just heard from Taiel Therapeutics as well as PTC Therapeutics developing small molecules. This is generally thought of as the traditional drug development process. But in personalized medicines, you're really trying to develop a much more targeted solution for mitochondrial disease patients. So Magali, maybe I'll start with you. Your company is developing a gene therapy. And I think most people in our audience today have heard of gene therapies or aware of gene therapies. Often they're positioned as cures, and as a part opposed to treatments. I think that's a conversation worth having. But maybe just in general terms, could you briefly speak to some of the challenges of bringing forward a biological solution like a gene therapy as opposed to a small molecule approach to drug development?
Sure. So I think gene therapy, most of the gene therapies actually treat the root cause of the disease firstly. So it's a very targeted therapy. It has advantages, but there are also some challenges, because when you want to treat, I would say, all mutations for all diseases, it takes time and a lot of development. Maybe one important advantage of gene therapy is it's a one-time treatment. Mm-hmm. So when you treat a patient, it's the promise of treating the patient for life with one injection. But it also brings challenges, because you have to show that the effect of a drug lasts over time. And there might be other challenges right now, because there are a lot of development of gene therapies, which is great for patients, but then you have the regulators who have to deal with that. And you have also the payers. So as soon as you promise to treat a patient with a one-time treatment, you need to have arguments with the payers and data to show that your treatment will last over time. And the big advantage one-time therapy, obviously, it's that you don't have any treatment observance issue, because it's a one-time treatment. On the other hand, what is important also to underline is it doesn't mean that you won't follow the patient. It's not because you do a treatment during one day and then you don't follow the patient. So the patient needs to understand he has to come back to be followed for safety, for efficacy, and so on. Right. So there are pros and cons, but in general, I think gene therapies constitute a great advances in the cure for patients with inherited diseases.
I agree, and it's very clear that going forward, we're going to see more gene therapy approaches to addressing rare inherited diseases. So thank you to Gensight for this early work in the LHON area and helping that portion of our disease community. Natalie, maybe you could just briefly speak to the cell therapy approach and contrast that with both gene therapy versus a traditional small molecule drug development.
Absolutely. So I would like to raise 2 points for maybe consideration that are very special for cell therapy, especially personalized cell therapy, such as what Minovia is developing. So first of all, a randomized control group is something that is very hard to have with such a special therapy. So as you know, Phil, we are mobilizing stem cells in drugs, and then we are conducting the apheresis process to collect the hematopoietic stem cells. So what is the control group in that regard, and whether it is ethical to bring patients for the full study and all the follow-ups without giving the therapy eventually, or giving just the placebo? That's something that we are still debating with the agency and with physicians, whether this is doable. So non-randomized control trials. So what is the control, and whether natural history can serve as control in that regard. So this is one. And the second element in personalized cell therapy is that your product is never the same, because you are counting on the cells coming from the patient. Every patient has a different, in case of Pearson, for example, a different deletion. And from every patient, you will have different numbers of cells, and you are actually injecting different number of cells based on the patient's body weight. And the controls are very important in terms of CMC and manufacturing. So this is a huge challenge for us for the development that is very different, I guess, from developing small molecules.
Yeah, for sure. But of course, the advantage would be, again, much like with the gene therapy discussion, if you're successful and the platform can deliver, then you really truly have a personalized therapeutic solution, right, for that mitochondrial disease patients. And that's a really exciting potential.
Sure, indeed.
Yeah, for sure. So thank you both for your comments on this, and obviously to Drs. Klein and Stacpoole as well. I think with this, we're going to end our Q&A period for this panel. I'd like to just complete this session by reminding our audience of a couple of things. One, all of the programming that you're seeing today and over the course of Mitochondrial Medicine 2021 is available 24/7 on demand through our virtual platform. And that programming will be available all the way through the end of July. I also want to mention, in particular to these clinical trial updates, many of the representatives that you see here today, or at least their organizations, are also participating in a networking session at the end of the day here on Friday, June 25th, in which the patient families will have an opportunity to directly interact with drug development companies. And this is important for a number of reasons, but my pitch to the patient community, this is an opportunity to really share your voice, what's important to you, what your priorities are, be able to ask questions of the representatives of the companies, but also listen and understand a little bit of the drug development process too. So I encourage all the patients, family members that are watching today live to consider joining the networking sessions at the end of the day. There will be multiple rooms and you're able to move between the rooms freely, no obligation to stay in any one room. And I think you'll find it really interesting. I know the company representatives are really keen to hear from you, so please take advantage of this opportunity. All right, with that, I'm going to draw to close this panel 1 of our clinical trial updates on mitochondrial disease, and finish by just thanking all of the panelists for the great updates and for the discussion, and look forward to hearing more in the future.
After this short break, our clinical trials panel discussion continues with representatives from Abliva, Baylor College of Medicine, Cyclerion Therapeutics, and Chondrion.